Nucleus and chromatin structure. Structure and function of nucleus in eukaryotes. The nucleus is one of the most important organelles in eukaryotic cells. It houses genetic material, DNA, and is responsible for controlling various cellular activities such as growth, metabolism, and reproduction. In this section, we will explore the structure and function of the nucleus and its chromatin in detail. Structure of nucleus in eukaryotes. Nuclear envelope. The nucleus is bounded by a double membrane known as the nuclear envelope. The outer membrane is continuous with the endoplasmic reticulum and may have ribosomes attached to it. The inner membrane provides structural support to the nucleus and interacts with nuclear components. The space between the two membranes is called the perinuclear space. Nuclear pores. The nuclear envelope is perforated by nuclear pores that allow the selective transport of molecules between the nucleus and the cytoplasm. These pores are regulated by the nuclear pore complex, NPC, which controls the movement of RNA, proteins, and other macromolecules. Nucleoplasm, nuclear sap. The nucleoplasm or nuclear sap is the fluid matrix inside the nucleus. It contains ions, enzymes, and nucleotides necessary for DNA and RNA synthesis. Nucleolus. The nucleolus is a dense region within the nucleus where ribosomal RNA, rRNA, synthesis and ribosome assembly occur. It contains DNA, RNA, and proteins. Chromatin. Chromatin is the complex of DNA and histone proteins found within the nucleus. It exists in two forms: euchromatin, loosely packed and transcriptionally active DNA; heterochromatin, tightly packed and transcriptionally inactive DNA. Chromatin structure. DNA double helix. Chromatin begins with the basic structure of DNA, which is a double-stranded helix of nucleotides. Nucleosomes. DNA wraps around histone proteins to form nucleosomes. A nucleosome consists of 146 base pairs of DNA wrapped around a core of 8 histone proteins, 2 each of H2A, H2B, H3, and H4. The histone H1 protein helps to link nucleosomes together, forming a beads-on-a-string structure. Chromatin fiber, 30 nanometers fiber. Nucleosomes are coiled and folded into a more compact structure known as the 30 nanometers chromatin fiber. This structure is stabilized by interactions between histones and DNA. Looped domains. Chromatin fibers are further organized into looped domains, which are attached to a protein scaffold within the nucleus. Condensed chromatin. Chromosomes. During cell division, chromatin further condenses to form chromosomes, ensuring proper segregation of genetic material. Functions of the nucleus. Genetic information storage. The nucleus stores the cell's genetic material, DNA, which contains the instructions for all cellular functions and hereditary information. Gene expression and regulation. The nucleus controls the process of transcription, where DNA is transcribed into mRNA. Regulatory mechanisms in the nucleus ensure that genes are expressed appropriately. Replication. DNA replication occurs in the nucleus before cell division, ensuring each daughter cell receives a complete set of genetic information. Ribosome biogenesis. The nucleolus within the nucleus is the site of ribosomal RNA, rRNA, synthesis and the initial assembly of ribosomes. RNA processing. Newly synthesized pre-mRNA undergoes modifications, including splicing, capping, and polyadenylation, before being transported to the cytoplasm. Conclusion. The nucleus is essential for the survival and proper functioning of eukaryotic cells. It serves as the control center, housing genetic material and coordinating various cellular processes. The chromatin structure ensures efficient packaging of DNA while allowing access to the genetic code for replication and transcription. Understanding the nucleus and chromatin provides insights into fundamental cellular mechanisms and how genetic information is managed within cells. Thank you for watching.